My father was a scientist, my mother was a GP, so I think that exposed me to science as well, actually. My father's research was in the area of metallurgy, which is material sciences, but he used electron microscopy to understand the structure of metals. So it's quite interesting that I'm now also using electron microscopy in my research to understand the structure of proteins. My name is David Barford. I'm a group leader in the Division of Structural Studies at the LMB. Until October, I was also joint head of division. I was an undergraduate at the University of Bristol studying biochemistry. I then moved to Oxford University for my DPhil with Louise Johnson as my supervisor. From there I moved to Dundee University for 1 year as a postdoc, and then I moved to Cold Spring Harbor Laboratory in New York State for a Cold Spring Harbor Laboratory Fellowship where I spent 3 years. I then returned to Oxford University as a lecturer and also a fellow of Somerville College for 5 years actually. So I left after 5 years to move to the Institute of Cancer Research in London, which was a purely research institution, which gave me more time to focus on research. And in 2013 I moved to the LMB as a group leader and then became the head of division in 2015. My research interests are focused on understanding how chromosomes are separated during cell division. So we studied the structures of protein complexes and machines which are involved in mediating and regulating cell cycle processes and also chromosome segregation at mitosis. So we use primarily electron microscopy to understand the structures of these complexes and how they organize the segregation of chromosomes during metaphase and anaphase transition. So kinetochores are large megadosome complexes which actually interact with chromosomes at specific sites on the chromosome called centromeres. They then attach the centromere to the microtubules, which are part of the mitotic spindle. The mitotic spindle then pulls the chromosomes apart during mitosis. Therefore, the daughter cells, which are then generated from the division of the mother cell, inherit the same number of chromosomes as the mother cell did initially have. So we want to understand the basis for the structures of these molecular machines, but also how they're regulated, how they actually know that all chromosomes are actually attached to the mitotic spindle. So when you separate the chromosomes, you need to ensure that all chromosomes are synchronized in their attachment. So when you do trigger anaphase, which is the process by which chromosomes separate, all chromosomes are separated and are then the daughter cells then inherit the same number of chromosomes. So that prevents aneuploidy, whereby you have different chromosome numbers in the daughter cells. You want to prevent that happening. Therefore, you have this very quite complicated regulated mechanisms in the cell which are controlling the attachment of microtubules to the chromosomes and also sensing tension, which implies that you have this biorientation of the sister-chromatid pairs, so that when you do separate, both individual chromatids of the pair are moved to the opposite poles of the cell, which ensures this polyploidy is correctly achieved. What inspired me to do this research? Well, I was interested in the structure of proteins as undergraduate, so I was fascinated by how proteins fold and understanding the actually protein folding question. How does a primary amino acid sequence give rise to a three-dimensional structure? There's still actually an unknown answer actually. So although AlphaFold can predict structures of proteins, it doesn't actually understand the sort of physical chemistry behind protein folding. So I always became interested in the structure of proteins and how they work and how they actually mediate cell functions. So that's my first sort of scientific interest. That's why I did a PhD in structural biology and crystallography actually with Louise Johnson in Oxford. And then the PhD project was focused on understanding how proteins are controlled by signals from outside the cell actually. So I became interested in understanding how signaling events are exerted through protein and change of conformation of proteins. So we understand how phosphorylation can activate a protein or can also inhibit protein. But so the question was how our proteins regulate and how they regulate other cell processes. And I thought, well, what's a very actually complicated cell process? And a complicated cell process is actually cell division, actually, because cells undergo very dramatic and profound changes of conformation and morphology during cell division. And it has to be really highly coordinated. You have to make sure you actually replicate DNA first, and then you have to separate the chromosomes. You have to have this ordered process of cell cycle events. So clearly a lot of signaling events are taking place in the cell cycle, and also that's actually where how many cancers occur by aberrant cell proliferation. So understanding the cell cycle from a molecular point of view is intrinsically very interesting, but also has this very important medical application for trying to treat cancer. And then I thought that chromosome segregation is a particularly interesting question, actually, because chromosomes are huge molecules in cells and have to be separated correctly. And but we didn't really understand all the machinery which was involved in chromosome separation. So I started my research on the question of chromosome separation probably 25 years ago now, when I moved to the Institute of Cancer Research from Oxford, because I moved there partly in order to really focus on this very long-term project and be free of teaching. Teaching can be interesting, but it's also a bit of distraction, frankly. So I wanted to focus on doing 100% research. So we worked initially on a complex called the anaphase promoting complex, which is involved in promoting anaphase, as the name suggests. It's also a large complex, 1.2 megadalton in size, and it's involved in triggering the first event of anaphase, whereby cystochromatins separate, and that's because the APC triggers the activation of a enzyme called separase, which then cleaves the cohesin, which is involved in cohesive assembly of cystochromatins. And then when I moved to the LMB, actually, 2013, we quickly actually determined the structure of the APC because of the revolution in electron microscopy. So we moved on very quickly, and we solved the structures of all the APC complexes within 3 years, so much faster than I expected, actually. And then we thought, well, next question is what are the other molecular machines which are involved in chromosome segregation? In fact, what does the APC do in order for chromosomes to separate? And that's actually then to allow the microtubules to pull apart the cystochromatins, which is the role of the kinetochores, to actually attach the chromosome to the microtubules, and also to actually harness the energy or power of microtubule depolymerization to move to opposite poles of the cell. So that's what brought me into structural biology, and then into cell cycle, and into chromosome segregation. The most exciting finding recently of our research has been the understanding of how the kinetochore actually interacts with the DNA. So the role of the kinetochore is to attach chromosomes to microtubules and to withstand the pulling and pushing forces during mitosis, because during mitosis microtubules are pulling on the chromosome, and you don't want the microtubule to sort of let loose of the chromosome during the pulling process. So how does the kinetochore act as its load-bearing element to ensure that it remains attached to the chromosome during this mitotic event? And what we discovered was that, well, it's known prior to work that the centromeres, which are the loci on the chromosome which binds to the kinetochores, which then attaches them to the microtubules, it's known that the centromere is defined by a specific nucleosome called the Sanpei nucleosome, which is different from the canonical H3 nucleosome. So people thought that the way that the kinetochore interacts with the centromere is by recognizing the Sanpei nucleosome. That would be the sort of anchor point by which the kinetochore would interact with the centromere. But we found, in fact, that the way that the kinetochore binds to the chromosome is simply by actually interacting with the DNA. DNA actually is passed through a tunnel through the entire kinetochore complex. So the complex actually surrounds the DNA like a hand holding a rope, actually. So you can see how that would actually provide a very strong, robust sort of load-bearing structure to allow the microtubule to pull on the kinetochore, but also remain attached to the DNA, single DNA duplex. So that was a surprising result we had recently. So the pivotal development, I think, in research recently has been the revolution in electron microscopy, which is driven by scientists here, Richard Henderson and also Shaw Sherris. That took place in 2013. That's one of the main reasons why I moved to the LMB, was to actually sort of take the opportunity of using this sort of revolution in electron microscopy to understand the structure of proteins. So this method allows us to understand the structure of very large complexes, which is very dynamic and actually unable to crystallize. So prior to electron microscopy, the technique to determine protein structure was to crystallize it. But that's actually very difficult when you have very large or very low abundance complex, which is often very dynamic. Whereas electron microscopy does not, of course, require crystallization, so you can actually look at single particles and they determine all the different heterogeneity in conformation and also in composition of these complexes. So that really did push my research forward dramatically, and it allowed us to determine the structure of the APC complex, which I mentioned earlier, very quickly. We thought we might spend 10 or more years on this project, but in actuality we finished everything in about 3 years. So that was a huge development in structural biology. Being here is really beneficial to doing the excellent science because of the interaction between colleagues and scientists, not just at the level of the group leader, but also particularly actually at postdoc and the student level. They're very, very interactive. So having all the shared facilities like electron microscopy, crystallography, biophysics, really fosters this sort of cooperative, collegiate atmosphere at the LMB. Grateful for that. We have this sort of opportunity to do full-time research. Actually, it's a really great privilege to be in a place where we just do research and pursue our interests, which are hopefully will lead to important discoveries and applications. And then we have the core funding. So we have, of course, that requires 5 yearly reviews, but it means we have a solid sort of support from the MRC for our staff, our groups, and also facilities. So NMB's been really very generous in terms of funding cryo-electron microscopes here. Actually, we've got really the best in the country by far, and we're very fortunate to have this facility. So, and then also the diversity of science here, and there's sort of great, fantastic colleagues and very, very clever people here. Actually, it's very clear that people here are really at the top of their field, and that's very obvious. And we benefit from that. Actually, we benefit from the sort of being around very able and clever people. Yes, what makes a good scientist? Well, first of all, you have to have curiosity. That's what drives you, because science is actually quite difficult. Although I say it's a job, it's also difficult. You have many disappointments and things don't work as you would like. So that's also why one should choose a really difficult and challenging project, because when it works, you realise you've made an important discovery. So it keeps you motivated to really sort of overcome some of the disappointments in science. But it also means you have to be persistent. You have to really sort of want to know the answer and then persist in tackling the question. You have to also, I think, focus. So one of the aspects of LMB is to focus on a few questions, not too many questions. So not be too broadly spread in terms of your interest. So you have to focus on some questions at the exclusion of other questions sometimes. So, but that means that you have time to focus and to concentrate on one or two things, and then. Yes, of course, you have to use good scientific approaches, which I was taught at school, actually. I remember now I was taught at school to do controls. That's a very important thing, is to make sure everything you do is controlled, so the outcome of the experiment actually is meaningful. Scientific breakthroughs in my own field, I would have liked to understand the rules for protein folding. That what actually inspired me early on in my career is to understand what are the protein folding rules, i.e. the second genetic code, which determines how protein folds into three-dimensional structure from a primary sequence. So AlphaFold actually was a program which actually can predict structures, doesn't explain how the amino acid sequence gives rise to the structure. So I think longer term you need to understand what are the basic physical rules which determine how proteins fold based upon their primary amino acid sequence. So I won't do that because I'm not in that field now, but I would like to have done so when I was younger, and I hope that someone will do it here. So hopefully we've appointed Joe Greener recently to work on this area, and so hopefully that the NMB will contribute to understanding the underlying physical principles which actually determine protein folding. In terms of my own research area, I would like to understand the structure of the kinetochores inside cells. So currently our approach is to reconstitute the kinetochore complexes in vitro, using overexpression systems and determine the structure of single particles, and then using cryo-electron microscopy. But we want to understand really the structure of the kinetochore bound to centromeres inside the cell. So the centromere of the human chromosome is about 2 megabases in length, and it's composed of 10,000 repeats of a 171 base pair repeat sequence. So that's a very sort of homogeneous repetitive structure. We know the structure of one of these repeats from our in vitro reconstitution experiments. We have no idea about how all these repeats are assembled inside the cell, actually at the chromosome. So to understand this, we need to use cryo-electron tomography, which is a means to visualize the structure of cells in their cellular context, so the structure of molecules in the cellular context. And that's our current research activity in the next 5 years, so we can make some progress towards that. I think what is unexpected is actually the transformation of science in the last sort of 25 years, actually, in 30 years, how technology is really sort of advanced to a point where we can do much more difficult science now than we could 25, 30 years ago. So that is unexpected, actually. So when I was doing my PhD, we had to do much more manual work in terms of mounting protein crystals into glass capillaries and then doing manual data collection. Even at synchrotron, you had to do manual using films to collect the X-ray data, manually sort of write on the film the number of the film, and then develop the films in the developer and fixer and dry them. But now everything's on electronic detectors and computers. So what's been the big change is how technology has really made a big impact on science. So that is what's not predictable actually, is how technology advances so much, and especially now machine learning and artificial intelligence, how that's having a big impact on science. So yeah, so 30 years ago, 35 years ago, I would not have dreamt that we would be able to sort of do things we can do now and do such large complexes and in vitro and also in situ. So that's been the big change, I think, or big unexpected development in my field. In terms of the breakthroughs in the next 5 to 10 years, in my field, I think the breakthroughs will be, as I mentioned before, our ability to visualize protein complexes in situ inside cells, so advances in cryo-electron tomography. So that will come from hardware development, so microscopes which can actually better visualize thicker specimens to visualize the machines in the cellular context to obtain better signal information from the tomograms. So that actually involves face plates to improve the information from the tomograms, and also CC corrected microscopes. This technique will also benefit from advances in computational methods to also use machine learning to try to interpret the tomographic data using existing information from structural biology or structural molecules of the machines inside cells. The second, I think, advance would be to understand the protein-protein question from a physical-chemical point of view. So how do proteins fold based upon the amino acid sequence? That's in the area of structural biology, which is also now interfaces with cell biology. Cell biology is also the question about the structure of cells, and that can be also addressed by using cryo-electron tomography. In cell biology, I think some questions which are important, which actually are sort of interrelated to structural biology, the structure of molecules inside cells and how they actually are dynamic. So everything's dynamic inside cells. And that can't be visualized really by using electron microscopy because we have to freeze the specimen. But one can visualize dynamic processes by using light microscopy. So very high resolution, both spatially and temporally, one allows us to understand the structure of molecules inside the cell and how they are changing their conformation or positioned inside the cell relative to other molecules. So I think understanding the dynamics of protein complexes in a high spatial and temporal resolution inside cells will be very important. That will probably develop in the next 5 to 10 years. I mentioned neuroscience and neurobiology. That will develop in the future because we'll be able to map the neurons in bigger organisms. So now we have the Drosophila brain just published recently, but hopefully we'll have the sort of mouse brain and eventually the human brain. So that will also be a huge advance in understanding neurobiology and memory and learning processes. My advice to a young scientist would be, if you're really curious about science, then you should do it. You have to have the curiosity to keep you going, to keep you motivated, and then to ask really difficult questions, and don't do the simple question, do the very challenging question. So that's, I think, the major prerequisites to succeed in science is be curious, but also accept there will be some disappointments, but that you overcome that by having the motivation to really address and answer a very challenging question. I think those are the main attributes to a young scientist to succeed, and hard work as well. Actually, one of my sort of scientific heroes was actually Dorothy Crowfoot Hodgkins, who was a crystallographer who worked on the structure of insulin and also penicillin and vitamin B12. She won the Nobel Prize for work on penicillin and vitamin B12. And she was at Oxford actually as a fellow of Solomon College. But she was apparently a very inspiring lady. I only met her a few times because she was retired by the time I was doing my PhD, but she did come for lunch a few times when I was having my lunch, and she was also very, very uplifting and encouraging and helpful actually. So I think I would like to work with her to understand how she managed to also raise a family, and also she didn't have a full-time job actually. She was actually appointed as a sort of fellow of Solomon College. She wasn't appointed by the University of Oxford for a long time, so she had to sort of juggle many sort of responsibilities as a scientist. But then she was very, very focused on. So I would think I would like to work with her. I think I also would like to work with Max Perutz, actually. So I met him a few times, and he was a very, very pleasant man, actually. Also very, 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 very intelligent person. So those two people I would like to work with. In my spare time, I enjoy going to theatre. So I enjoy going to London to watch plays at theatre and also to concerts. I enjoy reading, and also I enjoy walking. So those are my sort of pastimes.